Very good afternoon and salam sejahtera to all of you. Welcome to Immb International Webinar Series. My name is Hyo, and I'm your moderator today. And this afternoon, well, good afternoon to some of you from overseas. Yeah, good morning. So today we are really lucky to have Dr. Hiromu Kurahashi with us, a world-renowned fly taxonomist. He's going to share about his research and his experience in his whole career regarding the application of carrion flies, mango, dead body, and earth. And I think I found this very interesting, a message from flies to men. So during this presentation, please note that Dr. Kurahashi will present real time, but at the same time, we also will provide subtitle. Yeah, so you can look at the chat box or the message to look at the subtitle. All right. So without further ado, let me introduce Dr. Kurahashi, and it's our honor and pleasure to host Dr. Kurahashi in Immb, Faculty of Medicine, University Technology Mara, in Sungai Buloh, Malaysia. Dr. Hiromu Kurahashi, born on 1941 in Okazaki City, Aichi Prefecture, Japan. He is a distinguished entomologist specializing in taxonomy and medical entomology. He pursued his career with a Master of Science from Kanazawa University in Japan, followed by doctoral degrees in science from Tokyo Metropolitan University and in medical science from Tokyo Medical and Dental University.
Microphone out.
Dr. Kurahashi began his professional journey as an instructor and lecturer in the Department of Anatomy and Kanazawa University School of Medicine.
Microphone 1.
He then joined the National Institute of Health, later the National Institute of Infectious Diseases, where he served as entomologist in the Department of Medical Entomology. And over his career, he became head of the Taxonomy and Ecology Laboratory. And his main interest is Dipterology. Currently, he also lectures at Toyama Medical and Pharmaceutical University in the Department of Medical Zoology. After retiring from government service in 2002, Dr. Kurahashi continued his research as a fellow and currently heads the International Department of Dipterology. His lifelong contribution to insect taxonomy and ecology have made him a leading authority in his field, widely respected for advancing medical entomology in Japan as well as internationally. So it's our pleasure to host Dr. Kurahashi physically in this seminar. For this reason, I would like to invite Dr. Kurahashi to share his presentation. Thank you.
Thank you very much for the kind invitation and introduced me, Dr. Hyo-chan Chen. Thank you very much. And this is a very happy opportunity to show the message from flies to our human beings. Then the title is the application of carrion flies. So my name is Hiromu Kurahashi, and nickname the Happy Flies. Happy Flies. And, more than 30 years I studied flies. And now I learned much about the nature from the flies. So I am the entomologist, the taxonomist, medical and forensic importance flies. I was boy, I liked collecting insects. Boy, when I was born, collecting insects. I have collected many flies, made a large number of fly collection since the 1960s. I graduated from the Department of Biology, Kanagawa University, which is located in the Kanagawa city on the Japan Sea coast side of central Japan. I learned the taxonomy and biology of flies under the supervision of Professor Katsushige Hori for 7 years. And then I got a job as the research staff of the Department of Medical Entomology, National Institute of Infectious Disease, NIID. Formerly National Institute of Health, NIH. Now the name that changed. And continue to study the flight of medical importance, but I already retired from NIID at the end of the fiscal year, the 2000. So after that, already 25 years. I still continue to study the flies. So slide 2, please. これでいきますかね。Next one. This is a field work in the tropical rainforest. This is my field work style. Next, please. This is the National Institute of Infectious Disease, NIID in Tokyo, near the Waseda University. Okay, next, please. This is the slide 2. The adult flies. This is this shows the adult flies. Malayan. Morphologically, 2 wings. You can see the 2 wings. Only 2. They have the only 2 wings. A pair of wings. Almost all winged insects, except of fly, have 2 pair of wings. You know the morphology of the dragonfly, the butterfly, they have the 2 pairs of wings. But the fly Diptera, the 2-winged insect. And this is a very characteristic of the flies and the mosquitoes. And this best the hind wings. You can see the halter. This is the hind wings. Vestigial now is called a halter, and keeps a balance and the orientation of body while they are flying. Adult flies frequent decaying matter, such as dead animals and fishes. They also visit flowers for nutrition. The mostly a male flies like the flowers and visit to collect the eat the honey of flowers for grow the testes for making the and some kind of the vitamin that they need for the mature of the semi lava flies. Next, please. The lava flies are so that home one the adult. This is a lava, same species, but the larval stages like this. In English, maggot. Larval flies are called maggot in English. They are simple in structure. Structure very simple. like the bamboo, bamboo shoot. They have the no visible appendage, no legs, visible appendage, and eyes. Eyes also invisible. Larval, most species are scavenger and predators and parasitic in other insects and small animals. Scavenger means the environment. Scavenger flies play an important role of recycle of the resources, especially protein and ecosystem. Please, next, mango. This is a mango. Mango is well known as one of the delicious tropical fruits and very common in tropical countries. In Malaysia, you have the good mangoes everywhere. But mango is very expensive in Japan. We can buy the imported one from tropical countries. However, now in Okinawa, southern part of southmost island of Japan, farmers try to, next one please, grow the mango in the greenhouse. This is the greenhouse, produce a high quality mango. Its cost of one fruit are about more than 3,000 yen. Approximately is equal to the 100 ringgit, Malaysian ringgit. Very expensive. Farmers know the blowflies are good pollinator for mango flower. The oriental rattling flies, pollinator is the oriental rattling flies, Chrysomya, megacephala, and Chrysomya pingis are used. Farmers use these two species. These two species are very active in the greenhouse in winter. They are always requested to produce a delicious fruit of mango in Okinawa. Blowflies are a good pollinator for many kinds of wild and cultivated plants. It is well known that the Rafflesia bunga pakpa and a wild orchid and pollinated by the carrion breeding flies such as Sarcophagidae, Sensuratus, and Chrysomya. Next, please. This is the Chrysomya megacephala, the female, Obi-kimbai in the Japanese. This is the Rafflesia, the very strange flies. No stem, no leaf, only flowers like this, open. But pollinator is a carrion flies. Rafflesia male and female flowers grow in the different, far from each other. So between the male and female flowers are connected by the pollinators, the blowfly. Okay, next please. Next please. This also Sarcophaga sensuratus, this new species before the good pollinators, only endemic in the Malaysia. Very beautiful sarcophagus, fresh flies. The tail, abdomen, the tip of the abdomen are golden in the color. They're very characteristic forest species. The genus are different. Okay, next please. Now we move to the application, the Carrion fly to the forensic entomology, justice scientists. Dead body, carrion, carrion, dead body. Next, please. The carrion flies breathe in the dead bodies of the animal. In some cases, the dung too. The larval stages breathe in the dead bodies of animals. The next, please. This is a novel about the forensic entomological blowflies by very famous novel, black fly, blowflies. Next, please. This is the forensic and the traditional the forensic entomologies. And the first step of the forensic entomology is we must study the species of forensic importance in the area, the areas. The forensic entomologists should design the field experiment to know the entire fly fauna of the area. In the second step, the forensic entomologist also should design the laboratory experiment to know the growth rate of the immature stage of important species. Growth rate of the rabbit varies in the accordance with the temperature and the species. A growth curve must be calculated by using the good laboratory facilities such as Biotron. This slide 6 shows in the third step, if it is possible, entomological observation and correction should be conducted by the forensic entomologist at the death scene. Samples should be carefully collected, recorded, and identified. In the case of a no taxonomist, in your laboratory, please ask the specialist of each group of collected insects. No specialist, please contact the specialist of the group of the collected insects. Next, seventh slide. Slide 7, please. This is a sample, identification service of forensic importance flies and reference museum, Department of Medical Entomology, NIIDs. This is the whole application form and report, the result of identification. Left are the application and the right are the result of the identification. This is one of the case. Okay, next, please. This is a case. The left side is the newspaper that to 3 persons that died in the car in near the Tokyo. And the forensic entomology staff studies of this case. The Department of Tropical Disease and Parasitology, Tokyo University School of Medicine. And another one, the group of them in the Carrion, the network. these two are our home to request the identification of samples collected on the death scene and the report of the result identification service. Slide 8, please. And okay, 8. This slide shows the newspaper the reporting the death of 3 persons in the car in one mountainous areas. Slide 9, please. This is the picture that shows a flies emerges from the collected maggot on these 3 corpus. Chrysomia pingis, Lucia sericata, Becherisca peregrina were emerged from the corpus. PMI, in this case, will agree with the time of death. Okay, next, please. This is another view of the point of the carrion flies. Earth. On the earth, the Diptera, 2-winged flies, there were more than 100,000 species. Biodiversity, a big group of insects. Next, please, slide 10, please, finish. More than 100,000 species of Diptera has been described. Many new species are waiting for the description. It means the registration of the species in taxonomy. Most people, except me, don't like flies, especially maggot. Maggot, nobody likes the maggot. People think the flies are the harmful insect or pest, but most people hate flies. After 40 years study on flies, I became to love flies and hear the message from them. This is another viewpoint of the carrion flies. Slide 11. Slide 11. This is this slide shows a fly on feces. Feces, but it is not a real one. This is a mascot strap, which young Japanese girls like to wear. In Japan, the word means a good fortune. It has some pronunciation as the fishes. It's the same pronunciation, the fishes. So young girls like to wear this mascot, praying for their good fortune. This is my colleague's friend designed. I heard from my colleagues that 100,000 pieces were sold out at the cost of 100 yen a piece. This designer made more money, or 10 million yens in total, approximately 30,000 Malaysian ringgit. Another one, another one, please, next, please. This one. This one, my colleague, another colleague bought this topic, magnet stationary. Bought at the Carrion International Airport. Kuala Lumpur International Airport, this magnet. This is a more realistic, the Oriental rattling flies, design. Chrysomia megacephala in feeding on the piece of biscuit. Flies frequently come on food just after fed on the fishes. So in the case of fishes infected, in the case of the feces infected, we must keep all flies from the food to prevent infectious disease. This is a medically important point of view. Flies. But the flies, however, not always become dangerous. Transmission of infectious disease by the fly sometimes occur under the combination of the special situation. Next, please, slide 12. I'm afraid. Today, I would like to introduce another aspect of flies. Recently, I realized the flies are of great important creatures on the earth. Most species of carrion breeding flies due to their role in the decomposition and the recycling natural resources. Carrion breeding flies prey as scavengers on the earth. This is another aspect. Slide 13, please. We can easily understand they are skillful in the decomposition carrion or excrement if you observe the life cycle of flies. This is the life cycle of flies. Emerged adult, emerged the pupa emerged adult, juvenile young adult, adult, and matured the top, and they egg the hatchet, the rabbit, the pupa. We can easily distinguish. Hatchet rabbits very quickly feed on these material and decompose them soon. Scavenger flies are huge in the number of species, individuals. I already told you the number of fly species are more than 100,000 on the earth, for surrounding the quickly kept clean. Okay, next please, slide 14. This is common house flies. Today, I introduce you one of the excellent scavenger flies. It is a special strain of the house flies, Musca domestica, which was selected and improved by the project team of the Space Development Project in USSR, the former Soviet Union. This race of the house flies has strong power to decompose the animal feces and the dead bodies. Now, certain Japanese companies bought patent to use these flies. These companies continue to study these flies and try to make an industrial plant to decompose a large amount of wasted feces of domestic animals and the government program of fly project. Fly maggot reared from the feces are good food of chickens. Nutritious matter contained the feces can be obtained and recycled by the fly maggots. I tried to eat the grilled chicken, which were reared by the feeding fly maggot. These grilled chickens were very delicious. Okay, next slide 15, please. A large number of the chickens in a big farm, big farm, they excrete a large amount of the feces every day. the treatment of fish is a big problem of the environmental pollution. Can you understand the fishes become chicken? Can you understand the fishes became chicken again? How about your question? Answer. Yes, it is possible. If there are scavenger flies present in the ecosystem, nutritious matter in the fishes or dead bodies can be recycled by the presence of the scavenger flies. Actually, nature has this system and keeps her environment always clean and recycle nutrition material before complete chemical decomposition. Next, the chicken, the fishes, chicken question, but between the fishes and chicken, carrion flies, cycles. Next, please. 16, slide 16. After I studied the flies, I learned much from flies and finally realized that there is no simple useless species in the world, and believed there is no useless matter in the world. I found the similar word of light in the famous Japanese book Truth of Life, written by the Dr. Masaharu Taniguchi, where I found the meaningful word. There's no single useless experience in our life. Okay, next, please. The slide 17, the last. Okay, next, please. People say the 21st century is a century of environmental revolution. New economists point out nature has an infinite value within intact condition. Intact condition. Flies are always our friend, and even in the future, when we get an opportunity to live in the space station or other planet than the Earth. Thank you very much. This is my last. If all mankind were to disappear tomorrow, the world would regenerate back to the rich state of equilibrium that existed 10,000 years ago. If insects were to vanish, the terrestrial environment would collapse into chaos. This is my colleague, Dr. Pont, dung flies specialist from in 2007, personal communication. This is the last. Thank you very much for your audience. Thank you very much.
Thank you very much, Dr. Hiromu Kurahashi, for your sharing. Now we have some time for question and answers. So for the online participants, if you have any questions, you may type and write your questions in the messages or in the chat box. And I will try to read it for you. And we would like to later invite Dr. Kurahashi to answer your questions. So please, if you have any question, you may type in the chat box. And also, we have participants and audience in the seminar room. If you have any question, you may also raise up your hand and ask the question directly to Sensei. Yeah. Now we may invite questions from the participants. Okay. Yeah, please.
Yes, question, but I'm just wondering what is fly called in Japanese?
The question is how to call flies in Japanese.
Hiromu grow. Dead body. And after disappeared, like the grass. So this is the original.
Okay, we have one more question from online. Yeah, Zuhimi Mama Ismail. He said, "I'm conducting a research on carrion fly diversity, and I'm facing difficulties in pinning small flies for photography and keeping them as specimens. There's a method to take pictures under a microscope, but is there any other way to take pictures that look more natural?" That's the question.
I think we need a new technique, technique by using the photograph and the molecular analysis.
Yeah.
And but I think that this and forensic analysis more observation in the field, but not only also the outside of the field and take picture.
Yeah. The
Field work.
that would be the most natural. All right. Okay. Any question from the online audience? We still have time. And any question from our physical audience as well? Yeah, please, Asmira, please.
So when this is an unknown flies that you know, what are the first thing that you look at?
When you have like an unknown fly, unknown species, what are the first thing that you look at to identify?
Sorry.
じゃあ訳してもらえますか？訳しても大丈夫ですか？訳通訳します。先になんか密集がわかんない場合は先に肺を何を見ますか？
種かわからない。This is actually the problem. You mean the before the identification, you experiment the molecular. You can get that sequence, molecular identification first, and then to send the material specimens to the specialist in the taxonomy.
What structures from what structures we apply to identify to differentiate one fly from the other? How to differentiate two different flies?
How to?
First identify a fly. What you look at?
Good question. So we need the carrier. We need to many specimens that from the different and important. We need to see the type specimen. Type specimen is very important. This and the first year that this any species must be described morphological characteristic like this. This the material we call the type specimen. Type. This is the standard of the species. So we need to compare the my specimen and type specimen. This is the best way we know the exact name of the species. But it's very difficult to see the type specimen. Mostly in 1980 and 1758 the taxonomy started. Every animal or plant had scientific names. The 1758 was started. So after that was the type specimen description. But the most type specimen of Malaysian species, Malaysian fly. Type specimen preserved in the British Museum. Mosquito also. Other group of micro like this. Mostly the type of specimen in the British Museum. So they're very hard to see and the type specimen and compare to my specimen. Our taxonomy must be this kind of works. My specimen and the type specimen. But nowadays knowledge is better than the former. Easy to get the information. So nowadays we our specialist can identify the species as well as Japan and Malaysia also. Type specimen are best, but different specimen. Different specimen means that the specialist like me record the group of insects. Identify this is the Musca domestica and put the identification level on the specimen. This is the complete reference specimen. This is very useful for students for next generation of scientists. I think. So now I ask Dr. collaborate to make the difference correction of the carrion flies in Malaysia. Is it okay? This is the answer.
Okay, sister, thank you for the answer. I'd like to read for you the message from Dr. Martin Hall from the UK. He said, thank you for sharing your great enthusiasm for flies and your experiences, Dr. Kurahashi. It was a great pleasure to see you today. Best wishes from Martin Hall, Dr. Martin Hall. He was with Natural History Museum in London. Yeah. There's one question also from online from Ryan Larusi. He said, thank you for the talk. In my research, sorry, one moment, yeah. In my research, I collect Sarcophagidae larvae directly from bait, but in the larva mass, it's difficult to distinguish them from other fly larvae with the naked eye. Are there any practical field-level features or quick technique that you recommend for making more reliable identification during collection?
Me,
No, the Sarcophagidae, the larva. How do you differentiate the larva of Sarcophagidae from the other larva? Morphologically in the field.
This is a very first step of the forensic entomology. The many papers already published and about the larval stage, the first instar, second start, mostly the third one. So please refer that this previously published papers.
Yeah.
And please make a good series of the specimen. Rubber specimen are a little bit hard to make. We need a technique. Boiled, killed by the boiling water, and cut, clean the inside muscle or like that, and the skin, only skin, and pharyngeal skeleton, well, position. And the Diptera on the and
Right.
And if possible, some one batch lab for polymerization of the specimen. Another molecule.
Molecular, yeah. Sure.
So both now we can both. So the more
Right.
We don't need molecular identification. We know that. So you first install issue corpus, right?
Yeah.
Both morphological DNA, but especially the Malaysian species, studied and the other stage.
Yeah.
The molecular information of plastic. So then start. Good start.
Yeah. All right. Thank you for the answer. Yeah, I hope that answered your question, Ryan. Okay, there's one more comment from Olga Sivell, yeah, also from Natural History Museum in London. She said, "Thank you very much for your talk, Dr. Kurahashi. I'm a great admirer of your work, particularly on taxonomy. It's a great pleasure to be able to hear you talking in person. You're a role model and an inspiration, yeah, to many of us. Thank you for your brilliant work and many thanks to the organizer for the invitation." So thank you, Olga, for your kind message. All right. I believe we have one more message also online. Chairman, please convey this to Dr. Kurahashi. Sincerely great to know Sincere is teaching fly taxonomy to researchers in UITM. It will be useful for students to work on the DNA of these flies. Dr. Charles Lay from Sarawak.
Old friend.
Old friends. All right.
Thank you.
All right. Yeah, I know. Is there any more question from our physical audience anymore? Or any more question online? Also, please fill in the attendance form before we sign off. You can click the form, and this is for the e-certificate, right? Yeah, this e-certificate. One more message from him. Yeah. Okay. Perhaps you want to say any message to the audience? Any final message? Yeah. Right.
I have a more message from the Christ in our future that we have a big problem on the global warming and the environmental pollution. Then we need the this type right can survive in the cloud. Now, this morning I have received the email in Tokyo. Every day more than 40 degrees. So they are Tokyo now entered the boiling stage. Boiling. More than the body temperature, 36.5 higher. My blood boiling. But insects are no control with the blood and body temperatures. They have to depend upon the natural air temperatures. It could change like this, very hot and very cold. So the insect disappears. I continue to observe that along with my walking street from March this year. This year from March to now, flowers. Commonly, flowers, the many flies or the bees or the butterflies. But this year, from March to now, no insect on the flowers. No, not only flies, butterfly, whatever. So the this is my answer. So we need to the biodiversity, the natural environment, especially the tropical rainforest, very important to temperatures. Green, because you know the tropical rainforest, primary virgin forest. Temperatures 28 degrees almost all year. Every day. I forward the item. Put the maximum and minimum temperature. No, the thermometer. Every day that we do. Yes. So there, this is the the primary body temperature report. And then I ask, open the forest, no more trees. Temperature. So please carefully studies the change of the fauna. Urban areas and the rural areas and the primary forest.
Okay, thank you very much.
You're welcome.
Yeah, for the very important message. I believe what Sensei mentioned just now is an impact of climate change, environmental change that can affect our environment. Yeah, it will also increase the global temperature, and at the end will increase the population of pest insects, and at the end will lead to more infectious diseases. So in other words, we need to live in a harmony, equilibrium ecosystem that will be the sustainable future for humankind. So thank you very much, Sensei, for your message and your lecture and also your sharing. I believe many online and also physical audience share the same feeling like me and everyone here. We thank you for accepting our invitation to share your knowledge with us. Just as an information to all of you. Actually, there's one more question, from Zuhimi before I close. I feel like close it. What books, website, or journal that you always refer when doing the identification of these flies? Okay, there's one question from Mr. Zuhimi. Books or journals,
Identification.
Yeah.
The forensic information.
Or maybe any of carrion flies. I think what he studies is carrion flies.
Many books.
Yeah.
Then books of come to key family of is general text and theology.
Yeah.
And the Parisian Carrion fly,
Reference bulletin. Yeah.
In Malaysia.
Yeah.
What is the down fly? Muscidae.
Yeah. Okay. Sure. All right. I hope this answer you, Johemi. Yeah, you can refer to Sensei Dr. Kurahashi taxonomy key for the blowflies, flesh flies in Malaysia. Yeah, I think it's available online. So you can email us, or you can, for any other more questions, you can email Dr. Kurahashi directly. I believe the email address, we will later share in the already share in the chat box. So email any question you can please contact Dr. Kurahashi. And just to let you know that Dr. Kurahashi will also conduct a physical workshop on fly taxonomy this coming Thursday in Immb, Faculty of Medicine, University Technology Mara, Sungai Buloh, Malaysia. All right. So I think that's the end because of our time. On behalf of the Immb, our director here, Dr. Wang Seok is here. And the Faculty of Medicine, we would like to close this webinar, international webinar. If you miss it or you would like to share with your colleagues, I think this webinar is recorded. So later on, this webinar will be uploaded in our channel. Stay tuned for the recorded video. So with that, once again, thank you very much to Dr. Kurahashi. Thanks to audience, either you are physical or online. Thank you very much. Take care and stay safe, and hope to see you again. Thank you. Bye-bye.